So we're going to finish cardiovascular system by talking about blood vessels and blood pressure. But again, we are going to talk about blood pressure regulation when we talk about the urinary system, because your kidneys play a major role in blood pressure regulation. And then we're going to talk about blood in general when we talk about the respiratory system, because the whole point of the respiratory system is to get oxygen into your bloodstream and CO2 out. Okay, so this is not the last time we're going to talk about the cardiovascular system. It's the last major lecture solely about the cardiovascular system. Right, and before we talk about all those blood vessels and blood pressure regulation, we're going to do a little physics, but it's super easy physics. Okay, so we're going to talk about flow, pressure, and resistance relationships. And then we're going to march through the vascular tree. Okay, starting with the arteries, which drain into the arterioles, which drain into the capillaries, which converge into the veins, and then we'll talk about the accessory lymphatic system, and then finish off by talking about blood pressure. Not all today. Okay. So, the equation on the screen right here, flow rate, right, is equal to change in pressure over resistance. Is something you need to know, both for understanding blood flow, but also air flow. Okay, so we're going to see this equation again and talk about the relationship between flow rate and pressure and resistance when we talk about the respiratory system. Okay, so flow rate is directly proportional to changes in pressure. Okay, so the higher the pressure, the faster the flow rate. Where do you think the highest pressure readings are in the vascular tree? In the aorta, which is correct. Why do you think that's the highest pressure area? It's closest to the heart, which is the pump. Exactly. Okay, so you have fast flow rates occurring in the aorta. All right. Flow rate is inversely proportional to resistance. You can think of resistance as friction. Okay, and the smaller the blood vessel, the more friction blood encounters. So where do you think resistance is highest in the vascular tree? Capillaries. Okay, so arteries have really fast flow rates because they're under high pressure. Capillaries have really slow flow rates because there's tons of resistance. Okay, the pressure gradients, or delta pressure, is the pressure difference between the beginning and the end of a vessel. Fluids move from areas of high pressure to low pressure. Okay, so there's always higher pressure at the beginning of the vessel than the end of the vessel. Okay, resistance is a measure of opposition to blood flow. Okay, and it depends on three things: how viscous the blood is, how long the vessel is, and the radius of the vessel. The more viscous the blood is, the harder it is to push through the vascular tree. Okay, so that's why you warm up your syrup before you pour it on your pancakes, okay, to make it less viscous. Right? Easier to pour. You want your blood not to be super viscous. Okay, the longer the vessel, the more resistance, and the smaller the vessel, the more resistance. Okay, the radius of the vessel is the major determinant of resistance, because resistance, which is big R, is proportional to 1 over the radius to the 4th power. This means small changes in the radius lead to large changes in resistance. So here's some actual numbers for you guys to show you the effect of pressure changes on flow rate. So here we have two vessels, vessel 1 and vessel 2. They are the exact same size. Okay. The pressure difference in the start to end of vessel 1 is 40 millimeters of mercury. the pressure difference from the start to end of vessel 2 is 80 millimeters of mercury. Okay, so this means pressure in vessel 2 is 2 times that in vessel 1. So flow in vessel 2 is 2 times that in vessel 1. His flow rate is directly proportional to pressure changes. Okay, and the largest pressure differences occur in the arteries. Okay, so they have nice fast flow rates because they're under high pressure. Okay, resistance, the major determinant of resistance is the vessel radius. So here we have 2 different vessels. Vessel 1 has a radius that's half of vessel 2, or you can think vessel 2 has a radius that's twice that of vessel 1. So what's being shown up here is where 10 mils of blood, how much of that vessel it occupies. So in the smaller vessel, 10 mils of that blood occupies more of that vessel, right? And it's coming into more contact with the wall of that vessel. So it has more friction. Okay, so smaller vessels have more friction because more surface of the blood comes in contact with the vessel surface. Okay, this is why water flow is slower in creeks than in rivers, because more water comes in contact with creek beds, which are much smaller, than in larger river beds. Okay, so smaller vessels have higher friction, more resistance. So if we look at the actual numbers, the radius in vessel 2 is 2 times that of vessel 1. So resistance in vessel 2 is 1/16 that of vessel 1, because it's 1 over radius to the 4th power. 2 times 2 times 2 times 2 is 16. So this means the flow rate in vessel 2 is 16 times that in vessel 1. Small changes in radius lead to huge changes in flow rate. Okay, so small vessels have really slow flow rates. Okay, so here we have some figures showing average pressure in the vascular tree versus total cross-sectional area and mean linear velocity. And I couldn't stack them all up on the same column. Okay, but we have arteries, arterioles, capillaries, venules, and veins. Notice here we've got the systemic circuit and the pulmonary circuit. Huge difference in pressure, right? Pulmonary circuit is a much lower pressure circuit. Okay, which side of the heart pumps through the pulmonary circuit? Which ventricle? Right or left? Right ventricle pumps through the pulmonary circuit, and it has a much thinner wall than the left ventricle. Okay, so it can't develop as much pressure. Notice the arteries are highest in pressure. Huge drop off in pressure in the arterioles. Continues to decrease in the capillaries, venules, and then veins are under really low pressure. This is because blood is only going to flow down a pressure gradient. Okay, so in order for blood to return to the heart, those vessels supplying the heart with blood have to be under low pressure. Okay, because blood's only going to flow from high pressure to low pressure. So your veins are under low pressure. If we look here, though, at total cross-sectional area, you can think that this is inversely proportional to radius size or vessel size. So the more cross-sectional area of a vessel, that means the smaller the vessel is, the more you can pack in. Okay, so you have way more capillary cross-sectional area than aorta cross-sectional area. You just have one aorta and it's really large. If you open it up and lay it open, it actually doesn't take up that much space. Whereas if you were to open up all your capillaries and measure cross-sectional area, it would be 3,000 centimeters squared. Okay, because most of your cells are within a millimeter of a capillary bed, so you have lots of teeny tiny capillaries. Right, and then look at velocity. The smaller the vessels, the slower the velocity. And notice here in veins, even though they're under low pressure, so they're under really low pressure, they're really big, and therefore resistance drops, flow rate picks back up. Okay, so arteries have fast flow rates because they're under high pressure. Veins have fast flow rates because they're large and have very little resistance. You guys all fine with my physics? Right? So let's start talking about the various blood vessels then, and we'll start with the arteries. Okay, so our vascular tree is a closed system of vessels. Arteries carry blood away from the heart to tissues. Arterioles are branches of arteries. Capillaries are even smaller branches of the arterioles. They're the smallest vessels, and they're where exchange takes place. Our capillaries are leaky, right? And then capillaries rejoin to become venules, which are larger than capillaries. And then the venules converge in order to become the major veins. Veins return blood to the heart. Right, so here we have a cartoon showing the entire vascular tree. Obviously, it's highly simplified. So here we have a major artery that branches into arterioles, that branches into capillaries, that then converge into venules, that then converge into the major veins. Okay? The entire vascular tree is lined by endothelial cells. So we have endothelial cells in the inner portion of our arteries, arterioles. Our capillaries are only one cell layer thick, and that cell layer is the endothelial cell layer. Okay, and then we have endothelial cells lining the venules as well as the veins. Okay, and we have endothelial cells in our heart as well. So the entire vascular tree is lined by those endothelial cells. Okay, some blood vessels have smooth muscle. Arteries have a really thick, smooth muscle layer. Okay, veins also have a pretty thick, smooth muscle layer. Arteries' smooth muscle is not innervated. Veins' smooth muscle is innervated by the sympathetic nervous system. Okay, and that tells the veins to vasoconstrict. Arterioles, we don't have a nice cross-section showing the arterioles. Arterioles also have a smooth muscle layer that's innervated by the sympathetic nervous system. Okay, but that smooth muscle layer in the arteries is not innervated. Okay, and then we have a connective tissue layer. So in arteries, okay, that connective tissue has a lot of elastin. So arteries, in response to being stretched, recoil. And the reason they recoil is because they have a ton of elastin, which recoils in their connective tissue layer. Okay, so they want to recoil after being stretched. Veins also have a connective tissue layer, but they want it to be really stretchy, so they don't have as much elastin. Okay, so veins stretch and stay stretched. Veins are compliant. Right? And then veins also have valves. Okay, they're the only blood vessel that have valves, and they're one way. Because they're under such low pressure, if you didn't have valves, okay, all the blood would just pool up in your feet. Okay, so here we have average internal diameter and average wall thickness. So arteries have large internal diameters and are really thick walled, which is good because they're under super high pressure. Okay, you want a nice thick wall so that your artery doesn't dissect and then spill a ton of blood into the internal body compartment. Okay, they are highly muscular, highly elastic. They are not innervated. You can put that as a special feature as well. Not innervated. Okay, look at how much smaller arterioles are. They go from a 4 millimeter diameter vessel down to a 0.03 millimeter diameter vessel. Arterioles are where you get a major amount of resistance. Okay, they also have a smooth muscle layer, and they are well innervated by the sympathetic nervous system. Your arterioles are the major resistance vessels. They determine where your blood flow goes. Okay, capillaries are even tinier, internal diameter 0.008, extremely thin-walled because they're one cell layer thick, and they're leaky, they're permeable, they're where exchange takes place. They also have extremely slow flow rates. Okay. Capillaries will drain into much larger diameter venules, so you get this drop in resistance. Flow rate's going to start to pick up. Okay, they have some amount of smooth muscle. okay, but not a ton. You don't see a lot of smooth muscle until we get to the veins. And look at the veins. Internal diameter of 5 millimeters, even larger than your large arteries. Okay, so that increase in diameter drastically reduces resistance, flow rate picks back up. Okay, and they are compliant, so they can distend easily, and they have innervated smooth muscle, right? And then can vasoconstrict or vasodilate, depending on the body's needs. Okay, so arteries receive blood from the ventricles, and therefore are under high pressure. Because they're under high pressure, they have really fast flow rates. So that's the rapid transit passageway. Okay, they have high flow rates. Okay, and the whole point is to get blood from the heart into the arterioles, which then branch into the capillaries. Okay, they have very little resistance because of that large radius. The major role of your arteries is to act as pressure reservoirs. Because your heart is the pump. So it has to be the highest pressure point and the lowest pressure point in the vascular tree. Okay, your arteries maintain forward moving pressure in your vascular tree when your ventricles are in diastole. Okay, so when your ventricles fully relax, what is diastolic pressure on average in humans? So systolic pressure on average is 120. So diastolic pressure on average is 80. Okay, so arteries don't go down to 0. They only go to 80. So they maintain forward moving pressure so that you don't have your blood flow just coming in spits and starts whenever the artery or whenever the ventricles contract. Okay, because your arteries are under high pressure, they have to have a lot of connective tissue. Okay, that's going to contain collagen fibers to make it nice and strong, but they also need to be elastic so that they recoil. So there's a lot of elastin in your arterial walls as well. Okay, so here we have arteries acting as pressure reservoirs. So left ventricle, when it's in ventricular systole, okay, and doing ventricular ejection, when ventricular pressure is greater than arterial pressure, aortic valve opens, blood is ejected from the heart. That pushes against the walls of the arteries. Okay, as soon as the ventricles enter diastole, ventricular pressure is going to go below arterial pressure, aortic valve is going to close, and your arteries are going to recoil. And they recoil because of the elastic nature of the connective tissue. And then that really thick smooth muscle layer they have, one of the properties of smooth muscle is that in response to being stretched, it constricts. It's just an inherent property of smooth muscle. Okay? When we talked about the knee-jerk reflex, skeletal muscle doesn't inherently contract in response to being stretched. It actually has to have a stretch reflex in order to have it contract. Smooth muscle, if you stretch it, it's going to constrict in response. Okay, so that elastic recoil is due to the elastic nature of the arterial wall as well as the smooth muscle layer. Okay, and when your arteries recoil, that's going to push blood through the rest of the vascular tree. Right, and it also causes that dicrotic notch, right, in blood pressure readings, a little upward blip. Okay, any questions about arteries? Okay, arteries branch into arterioles, which are much smaller. Okay, and they're therefore called the major resistance vessels. So you go from an average of 4 millimeter internal diameter to an average of 0.08 millimeter internal diameter in the arterioles. So huge drop in the size means huge increase in resistance. Okay, and then the radius of your arterioles changes depending on your body's needs. Okay, they are innervated, and they're also sensitive to local chemical messengers, and they vasoconstrict or vasodilate in response. Okay? And they are a major determinant of overall blood pressure. So if all your arterioles vasoconstrict, you're going to see an increase in blood pressure. If they all vasodilate, you're going to see a decrease in blood pressure. Okay, so here we have an arteriole that's at rest. Here's the smooth muscle layer and the connective tissue layer. Okay, when this smooth muscle constricts, it decreases the internal diameter. Okay, that's going to increase resistance and slow blood flow. So any downstream tissue is going to get less blood. Okay, when the smooth muscle relaxes, that causes vasodilation, which increases the internal diameter, which will decrease resistance, which will increase flow rate, which will mean any downstream tissue is going to get more blood. Okay, so it acts like a spigot. Vasodilation is turning on the spigot, get more flow. Vasoconstriction is like turning off the spigot, less blood flow. Right, so what causes vasoconstriction? So this is what causes the smooth muscle layer to constrict. Right? Increase in myogenic activity means the smooth muscle is contracting. Okay, that's what causes vasoconstriction. This happens due to an increase in oxygen. Okay, so if downstream tissues are not active, right, they're not needing to make a lot of ATP, you're going to see a localized increase in oxygen in that area. And the smooth muscle in the arterioles can detect that, right, and they vasoconstrict in response because that tissue doesn't need as much blood. It's not active. Okay. Tissues that aren't active produce less carbon dioxide, right? Because they're doing less cellular respiration. They also need less blood flow, so you get vasoconstriction. Okay. Other metabolites like less hydrogen ions, etc., will cause vasoconstriction. Okay. The chemical messenger endothelin, produced by endothelial cells, will cause vasoconstriction. Right? And then these are all external things. Sympathetic stimulation, sympathetic nervous system innervates arterioles. It will cause vasoconstriction. The hormone vasopressin, also known as antidiuretic hormone, causes vasoconstriction. Okay? And then angiotensin II, which is part of the renin-angiotensin-aldosterone system, it also causes vasoconstriction. Okay. And then local application of cold causes vasoconstriction. Right? So when you go out in the cold, the blood vessels supplying your skin surface are going to vasoconstrict so you don't lose heat to the external environment. Or say you get a sports injury, okay, and you ice that area, it's going to cause vasoconstriction, which will decrease inflammation. Okay, vasodilation occurs when there's less myogenic activity, when that smooth muscle layer is relaxing. Okay, and this is due to decreases in oxygen or increases in carbon dioxide. So active tissues use the oxygen and produce CO2, and that tells arterioles supplying them to vasodilate. They need more blood. They need more nutrients and more oxygen. Okay, nitric oxide, NO, will cause vasodilation. Okay, it's produced in the lungs, and that will cause vasodilation. And then lack of sympathetic stimulation will cause vasodilation. Histamine release will cause vasodilation. It's part of the inflammatory response. And then local application of heat will cause vasodilation. Right, so your book has a pretty complete table, right? You need to know the effects of oxygen, and carbon dioxide, right? And then we talked about endothelin and nitric oxide, right? But remember how that list sometimes said oxygen, carbon dioxide, and other metabolites, things like potassium ions, acids, adenosine. That would be the other metabolites they were talking about. Okay, but just make sure you know what the role of oxygen, carbon dioxide, endothelin, and nitric oxide are on what they cause vasoconstriction or vasodilation. Okay, extrinsic controls. Whenever we talk about extrinsic, that means nervous system or endocrine system. Okay, intrinsic are local effects. Extrinsic is always the nervous system controlling something or the endocrine system controlling something in an organ. Right? So sympathetic nervous system causes vasoconstriction. Okay, epinephrine, depending on the receptor type, causes either vasoconstriction or vasodilation. The arterioles supplying your skeletal muscle express the beta-2 adrenergic receptors. Okay, so that's why you get vasodilation of those blood vessels during exercise. Okay, other blood vessels do the alpha adrenergic and get just vasoconstriction going on. Okay, vasopressin and angiotensin II, which are both hormones, cause vasoconstriction. Okay, notice most extrinsic control is vasoconstriction. Right, so here we have one last flow diagram looking at the effect of intrinsic versus extrinsic on arteriolar radius. Okay, so intrinsic is just the myogenic response. When it gets stretched, it's going to constrict in response. Here it's saying that heat or cold application is an intrinsic thing. Okay, so that's a hot compress or a cold compress. Histamine release. Right? And then extrinsic, we've got two hormones, antidiuretic hormone and angiotensin II, and then epinephrine and norepinephrine from the sympathetic nervous system. Notice down here, these are the major factors affecting the radius. Sympathetic activity in general causes vasoconstriction, right? But local metabolic changes always trump sympathetic. Okay, so local metabolic changes in oxygen, carbon dioxide, other metabolites will always trump sympathetic stimulation. Okay, so fight or flight, you'll cause system-wide vasoconstriction, and then tissues that are active will override it and cause vasodilation. So you actually don't see a huge increase in blood pressure overall. Right, because those blood vessels supplying your skeletal muscles vasodilates. Whereas if you just have fight or flight going on and no activity afterwards, such as in the testing center, right, blood pressure will skyrocket. Okay, here we also have blood viscosity. Okay, blood viscosity is dependent on the number of red blood cells and the concentration of plasma proteins. Okay, so this is why blood doping can potentially be dangerous. You get the number of red blood cells up too high, right? It makes the blood way more viscous and the heart has to work harder to pump it. Okay. Those arterioles determine where blood flow is going to go. So here we have a pie chart showing where cardiac output at rest goes versus exercising cardiac output. So cardiac output, this pie is smaller than this pie. This is a 5-liter pie. This is a 25-liter pie. Okay. The 5-liter pie at rest, right? Some goes to skeletal muscles, liter goes to kidneys, liter and a quarter goes to the GI tract, brain gets almost a liter, right? Kidneys, GI tract are conditioning organs, right? So they're getting a larger blood supply than they need for their own metabolic needs. GI tract is adding nutrients. Kidneys are filtering the blood. Okay, the heart's going to get a constant supply of blood because whether you're resting or exercising, it's working, right? Notice though, once you start to exercise, that massive increase in cardiac output, where does the biggest portion of it go? To skeletal muscle. It goes from 1 liter all the way up to 20 liters. Why does the skeletal muscle get all that extra blood? For energy, exactly. Contraction is energetically expensive. Lots of oxygen's needed, lots of nutrients are needed. Okay, notice your kidneys see a decrease in blood, as does the GI tract. Okay, but the heart sees an increase in blood. It goes from 0.2 to 1.15, because that's what's controlling cardiac output. It's actually working harder. Brain gets a little higher blood supply. Okay, but your brain is going to get a pretty constant supply of blood, whether you're exercising or not exercising. Okay, and then your skin sees an increase in blood supply. It goes from 0.25 to 0.65. Why do you get an increase in blood supply to your skin? Exactly, temperature regulation. Okay, your skin's not active. It's just helping regulate your temperature. Okay, so continuing on our trip through the vascular tree, we've now hit the capillaries, which are extremely thin-walled. They're only one cell layer thick. All they have is the endothelial cells. Okay, they have a really small radius, and they're heavily branched. Okay, since they have such a small radius and heavily branched, they have a huge amount of cross-sectional area. So the total cross-sectional area of your capillaries is about 600 meters squared. Okay, so if you cut them all open and laid them out. This means that most of your cells are within a millimeter of a capillary, because most of your cells are way too far away from air to get the oxygen that they need. Okay, so capillaries are delivering oxygen and they're delivering nutrients, and then they're transporting wastes away. Okay, so your capillaries are where you get exchange between blood and surrounding tissues. Okay, because they're so small, they have tons of resistance, so flow rate is really slow. Okay, flow rate in your capillaries is really slow, and that's a good thing for exchange. So here we actually have a micrograph of a capillary, and notice that your red blood cells have to squeeze through one at a time. Okay, it gives tons of time for exchange. Okay, exchange takes place in two different ways. One is diffusion and one is bulk flow. Diffusion is just molecules moving down their concentration gradients. So from where they're in higher concentration to lower. So oxygen is going to move out of the cell, out of the blood, into cells. CO2 is going to move into the bloodstream. Okay, the other one is bulk flow. Okay, so that's pushing fluid out of the capillary beds at the beginning of the capillaries, and then drawing water back in at the end of capillaries. And anything water soluble will move by bulk flow as well. Okay, we have two major types of capillaries. One is continuous and one is fenestrated. Continuous capillaries are the most common. These are the ones that are supplying most of your tissues. This is where you just have gaps between the endothelial cells. Okay, so you just have leaky areas in between the endothelial cells. Anything small and water soluble can leak out. Okay, in the blood-brain barrier, those continuous capillaries, those gaps, form tight junctions instead because of the astrocytes. Right? But most other tissues just have continuous capillaries with small gaps between the endothelial cells. Okay, some tissues have what are called fenestrated capillaries, which are way leaky. Okay, so these are the super leaky capillaries. So they have really big gaps between the endothelial cells. And they're found in tissues like your liver, kidneys, and bone marrow. Why do you think your liver has fenestrated capillaries? It does a lot of filtering, that's true, but is it adding anything to the blood? What makes most of your plasma proteins? The liver. Okay, so your liver makes most of your plasma proteins, so it has to be able to get them into your bloodstream. Okay, and your plasma proteins play a major role in holding water in the bloodstream. So people who go into liver failure often suffer from major edema or swelling of their tissues because they're not making their plasma proteins anymore, so their tissues all swell up because the proteins aren't pulling that water back in. Okay. Kidneys. So why do your kidneys have fenestrated capillaries? Same answer for liver. A lot of filtering. Okay, so those fenestrated capillaries are found specifically in the glomerulus. Okay, so the glomerulus is a capillary bed within your kidneys, within the nephron, that does a ton of filtration, and it has fenestrated capillaries. Okay, and then the bone marrow. Why does your bone marrow need fenestrated capillaries? What's your bone marrow making for you? Red blood cells, some white blood cells too. Okay, so they have to have big enough pores to get those blood cells into the bloodstream. Okay, so here we've got an example of a continuous capillary and a fenestrated capillary. So they're both only one cell layer thick. Okay, so they only have endothelial cells. Those endothelial cells are held together by a basement membrane. These basement membranes are considered an acellular layer. Okay, but they're keeping those endothelial cells together in a sheet. Right? Notice that we have a narrow intercellular gap in our continuous capillaries. Small water-soluble things can pass through that gap. Fenestrated capillaries, those gaps are much larger, so big things like what white blood cells can pass through. Okay, so here is how things move through normal capillary beds. So we already talked about how stuff moves through the blood-brain barrier, right? And those capillary cells have tight junctions between them, so everything has to cross through cells. In continuous capillaries that we find in other tissues, small water-soluble molecules like the ions and glucose and amino acids go through pores. Okay, so they're just going to move down concentration gradients, whether they're in higher concentration to lower concentration. Anything lipid soluble can cross through the endothelial cells. So oxygen, CO2, fatty acids, steroid hormones, okay, just goes through endothelial cells. Proteins are too large and remain behind. Okay, and if they are going to be moved to extracellular fluid, they're going to move by transcytosis. So remember how we talked about the clotting cascade? And there is the clotting cascade that happens within the bloodstream, and then there's clotting factors in your tissues, right? And do a second type of clotting cascade. Those clotting factors are made by the liver, and then they're put into the extracellular fluid, the interstitial fluid, via transcytosis. Okay, so we're going to talk about bulk flow. So this is the movement of water, right, from capillaries, so from plasma to interstitial fluid, and from interstitial fluid back into plasma. So remember, both are extracellular fluid compartments. Okay, so both are part of the ECF. Plasma is the extracellular fluid found within the circulatory system. And then interstitial fluid, which I'm abbreviating ISF, is the extracellular fluid bathing your tissues. Okay, so protein-free plasma moves out of the capillary beds, right? When it moves out of the capillaries, it's filtered. Okay, so filtration is movement from plasma to interstitial fluid. And then when it moves from interstitial fluid back into capillaries, it's called absorption. Okay, so when things move from interstitial fluid back to plasma, it's called absorption. You get net filtration at the beginning of capillary beds, so fluid gets pushed out at the beginning of capillary beds, and then you want net absorption at the end of capillary beds. Okay, so you don't push all the fluid out into your tissues and then they'll just swell up. Okay, the purpose of bulk flow is to distribute extracellular fluid. Right, to keep all your cells, both in your bloodstream as well as outside your bloodstream, happy and plump. Okay, bulk flow is due to hydrostatic and osmotic pressures. Okay, so water moves due to being pushed on, so that would be hydrostatic pressure. And then it also moves when it's pulled by solutes. That's the osmotic pressure. Okay, so hydrostatic pressure pushes fluids. Osmotic pressure pulls fluids. All right, so let's look at this figure. We have a really simplified capillary bed. We have the arteriole, the blood's oxygenated, that's why it's red. Okay, it's going to enter the capillary. Normally capillaries, all right, are going to be diffuse and branched. However, in this example, we just show one capillary in between the arteriole and the venule. Capillaries where exchange takes place. So this is where oxygen is going to be leaving the bloodstream. So that's why it's becoming purplish. And then venule is going to be returning the deoxygenated blood back to the heart. Okay, so here we've got a series of pressures. P is a hydrostatic pressure. Pi is osmotic pressure. Okay, so IF is interstitial fluid. Okay, capillary is hydrostatic fluid in the capillaries. The size of the arrows tells you the relative force. Okay, so it's a bigger area or a bigger arrow for the pressure in the capillary. So blood pressure inside your capillaries is greater than the hydrostatic pressure of your interstitial fluid. Okay, so capillary blood pressure is what causes net filtration at the beginning of the capillary bed. Okay, blood pressure pushes plasma out of the capillaries. Right? At the end of the capillary bed, it's all about osmotic pressure. And osmotic pressure is all about where the solutes are. Okay, so here we have the osmotic pressure of interstitial fluid. So the amount of solutes in your interstitial fluid pulling water to it. Okay, and here's osmotic pressure of the capillary. So those are all the plasma proteins remaining behind, pulling water towards them. And again, the arrows show relative strength of that pressure. And so osmotic pressure of the plasma is much greater than of the interstitial fluid. So that's going to draw water back into the capillary and cause net absorption at the end of the capillary bed. Okay, so capillary blood pressure pushes fluid out at the beginning of the capillary bed. Blood osmotic pressure pulls fluid back in at the end of the capillary bed. Okay. Yeah?
Is the filtration and absorption equivalent?
Almost. Pretty close. With all your capillary beds, you only lose about 2 liters a day that doesn't get returned at the end of capillary beds, which isn't so bad.
Okay.
Yep. And that gets returned by the lymphatic system. Okay? So it's pretty good. Right? So again, get net filtration at the beginning of the capillary bed, net absorption at the ends. Okay? And here's some numbers if you want to work through them. I won't make you calculate anything though. Okay. So, I said 2 liters. It's 3. About 3 liters, sorry you guys, about 3 liters of fluid that gets filtered out of all your capillary beds doesn't get returned at the end, right? But that 3 liters of fluid doesn't stay in your tissues. Otherwise, everything would be all swelled up or all swollen. Okay, it gets returned to the vascular system via the lymphatic system. Okay, and your lymphatic system is one-way vessels. Okay, they have their endpoints are in all of your capillary beds. They're blind-ended at your capillary beds. Okay, and then we're going to return all that excess filtered fluid into the bloodstream at the vena cava. Right? And the vena cava then dumps all your blood into the right side of your heart. Okay, so lymph flow is only 3 liters per day, whereas blood flow is 7,200 liters per day. Okay, so lymphatic system is a really low flow, low pressure circuit. Okay, it helps return excess filtered fluid. It also is filled with lymph nodes that have resident phagocytes, okay, that destroy any bacteria that get filtered out. So bacteria can enter your lymphatic system. Okay. And so those lymph nodes have resident white blood cells that are always screening the lymph for anything foreign. That's why when you get sick, sometimes your lymph nodes swell, right? Your lymphatic system transports absorbed fat from the GI tract. Okay, the way you package fat, when you absorb it, you package it into chylomicrons. Okay, you package it with protein. Because if you just dumped a bunch of fat into the bloodstream, what would happen? What does fat do in water? It doesn't dissolve, right? It's going to clump all together. You don't want a bunch of fat droplets circulating in your blood, right? So you package the fat you absorb from your GI tract into chylomicrons, so they're protein and fat, and they're too big to get into capillary beds. So instead, they enter the lymphatic system, okay, and then get dumped into the bloodstream at the vena cava. Right? And any protein that gets filtered out of the bloodstream also gets returned via the lymphatic system. Okay, so here we have the systemic circuit and the pulmonary circuit, and what's being shown is that you have lymphatic capillaries associated with your pulmonary circuit as well as your lymphatic circuit. Okay, you have a series of lymph nodes. It's just showing one in each, but you have loads of lymph nodes associated with both lymphatic and systemic circuit. Okay, and then you have one-way valves. Your lymphatic vessels have one-way valves because they're under super low pressure. Your veins also have one-way valves. Okay, those one-way valves prevent backflow. So low pressure vessels have one-way valves so that you're not going to have all that fluid just filter in lower or sit in lower extremities. Okay, and then both the lymphatic system associated with your pulmonary circuit and systemic circuit, they dump that lymph into your vena cava, so all that fluid and proteins get returned, and dietary fat get returned to the bloodstream. Okay, so the lymphatic system, we have these initial lymphatics, and that's where fluid and bacteria and protein enter. Okay, and they're blind-ended. They're the terminal lymph vessels associated with your capillary beds. Okay, lymph is the interstitial fluid that's in the lymphatic vessel. Okay, so it's a subcategory of interstitial fluid, which is a subcategory of extracellular fluid. Okay, so lymph is just the fluid inside your lymphatic system. Okay, your lymph vessels are formed from the convergence of those initial lymphatics. Then they eventually dump all that fluid and filtered protein into the vena cava. They've got one-way valves. Okay, so here we have some initial lymphatics being shown. Notice they're blind-ended. It's not a circuit. Okay, so here's the beginning of a capillary bed where fluid's being pushed out. End of capillary bed, fluid's being reabsorbed. Right? Any fluid that doesn't get back into the end of the capillary bed will enter the lymphatic system. And the reason that excess filtered fluid and proteins and bacteria, etc., can enter the lymphatic system when they can't get back into the capillary beds is that the endothelial cells that make up those initial lymphatics are overlapping. Okay, and what happens is when you have an increase in pressure due to a buildup of interstitial fluid, because too much fluid's been filtered out and not enough's been reabsorbed, that opens up flaps. And those flaps are really big, so big things can get in. Okay, so it's easier to get into an initial lymphatic than into a capillary bed. Okay, increase in pressure just opens up these really large fat flaps, right? And large things can get in, like chylomicrons, bacteria, filtered protein. Okay, I mentioned that if you push out too much fluid and you don't return enough of it, you can get what's called edema or swelling of tissues. Okay, so this is when too much interstitial fluid accumulates. And it's pretty common, right? Causes of edema can be reduced concentration of plasma proteins. This is what happens in liver failure. Yeah?
It's also kind of what happens when you get pregnant.
Yes. Exactly. So when you get pregnant, your feet swell especially. Okay, and all of you experience this if you go on a long airplane ride or if you stand on your feet all day long, right? You just don't return all of that excess filtered fluid in your lower extremities. Okay, and when you're pregnant, you got this giant thing that's affecting venous return from your lower extremities. Okay, if you get a boo-boo, okay, you may set off the inflammation response, and things like histamine will increase the permeability of your capillary wall. So even more fluid will leave, not all of it will get returned, and you get localized swelling at the site of injury. Right? Increase in venous pressure. This is what happens to your lower extremities. Okay? They're under super low pressure, but then gravity's pushing down on them, so it gets really hard to return that blood, especially if you're not using skeletal muscles. Okay, so your major veins in your lower extremities run through your major skeletal muscles in your lower extremities, and when you contract them, that helps to squeeze blood back towards your heart, because your veins are under super low pressure. Okay, the other one is blockage of lymph vessels. This is what happens in elephantitis, right? There's a parasite that lodges itself in the lymph vessels. We don't have it in the U.S., okay? And then whatever area of the body that parasite is lodged in just swells up like mad, right?
Is that, do you remember?
The one that causes elephantitis? No, I don't know. Yeah, it's the one that causes elephantitis. I don't know which parasite. Okay. Any questions about lymph system before we finish our last blood vessel? Okay, so now we have to return blood to the heart. That's going to be done by the veins. Okay, so capillaries drain into venules. Venules are larger in diameter to your capillaries, so flow rate picks up because resistance starts to decrease. Okay, venules converge to form the small veins. That's what exit your organs. And then the smaller veins converge to form the larger vessels. And each one gets larger and larger in diameter. Right? And that increase in diameter massively decreases resistance, so flow rate picks back up. Okay, because your veins are under low pressure. Okay, so flow rate picks back up in your veins because they're under low pressure. Your veins act as blood reservoirs. At any given time, 60% of your blood is in your veins. And the reason why your veins act as blood reservoirs is because they're really compliant. Compliant means stretchy. Okay? So if you're the type of person that's easily swayed, you could call yourself compliant. Right? It's a good vocabulary word. You can use it in physiology, but you can also use it in everyday life. Okay, so here we have the compliance. So here we have volume in the arteries versus veins under the same amount of pressure. So for any given pressure, your veins stretch way more than your arteries. Okay, so say this is a pressure of 10 millimeters of mercury. Okay, that doesn't stretch your arteries very much. They're elastic. They're not compliant. They're elastic. Okay, whereas your veins are super stretchy. Okay, they're compliant. They stretch easily and they stay stretched. Right, so here we have a pie chart showing where your blood is at any given time. Notice even though you have a huge amount of cross-sectional area for your capillaries, it only holds like 5% of your blood because they're so stinking small. Right? Your red blood cells have to squeeze through one at a time. Right? Your heart holds about 8%. Okay, pulmonary system, the whole pulmonary system only holds about 12%. Okay, and then your systemic arteries and arterioles hold another 15%. Most of it is in your veins and venules, okay, because they're so stretchy. Okay, veins are under super low pressure. Oh, sorry.
Oh, I was just going to say, so that compliance, is that why you get varicose veins? They just get bigger and bigger?
Yeah. So as you age, right, and then varicose veins are much more common in people who do a lot of standing, or they often occur during pregnancy, right, because you have increased venous pressure. Yeah, causes the varicose veins. Yep. Okay. So veins are under super low pressure, right? Because fluid only moves down a pressure gradient, so from areas of high pressure to low pressure. So since they're at the end of the vascular tree, they're under low pressure. So there's a series of things that help increase venous return, getting the blood back to the heart. Okay, one is the pressure from cardiac contraction. Okay, so every time during systole, that's providing a little bit of pressure to get blood to come back to the heart. Another is that your veins have a smooth muscle layer, and that smooth muscle is innervated by the sympathetic nervous system. And then the sympathetic nervous system causes that smooth muscle to vasoconstrict. And when it squeezes, there's valves in your veins, one-way valves. So when your veins vasoconstrict, blood only has one way to go, which is back to the heart. Okay, so the sympathetic nervous system can cause venous vasoconstriction as well as arteriole vasoconstriction. Okay, skeletal muscle activity. Most of your major veins run through major skeletal muscles, and when your skeletal muscles contract, that squeezes the veins. The one-way valves mean blood only returns to the heart. This is especially important in lower extremities. So this is why on long flights, you're supposed to get up and walk around, and that helps with venous return. Okay, mentioned valves multiple times. One way, only open to allow blood to return to the heart. Yeah?
I just wanted to share with you. About a month ago, there was a real well-known gamer that was online for 24 hours. They moved, he died because of heart failure.
Right, so what can happen is if you don't get up and get that blood returned to your heart, it can clot in your lower extremities. Okay, so stagnant blood flow can cause clotting, and then if that clot isn't in an injured area, it can dislodge. Okay, and if it lodges itself in your pulmonary circulation, you can get a pulmonary embolism, that can kill you. Or if it lodges in your coronary circulation, that will cause a heart attack, that can kill you. It can lodge in your brain, that can kill you, right? So don't be lazy. Get up and move around. Okay, the other thing that helps with venous return is respiratory activity. So every time you inhale, take a deep breath in, the volume of your thoracic cavity increases, pressure decreases. That helps to suck blood up to your thorax, which is where your heart is, right? Every time you exhale, that decreases the volume of your thorax, increases the pressure. So any blood that's already in your thorax only has one way to go because of one-way valves, which is into your heart. And when you exercise, what happens to respiratory activity? It increases. So exercise, that increased respiratory activity, helps increase venous return, right? Which helps increase cardiac output. And then the other thing is cardiac suction. Okay, so your ventricles are ultimately the highest pressure point and lowest pressure point. So at the end of diastole, they're almost at 0 millimeters of mercury. So they act like a vacuum and they suck blood into them. Okay. So here we've got the effect of skeletal muscle on venous return, as well as showing those one-way valves. Again, this is really important for returning blood from our lower extremities. Okay, so we have these one-way valves. Every time muscle contracts, squeezes the veins, there's only one way to go, which is back to the heart. Okay. Wearing compression socks does the same thing too. So people who spend a lot of time on their feet, right? Or sometimes if you do, you're going to do a long flight, you put on the really tight compression socks, okay? And that helps with venous return as well. Okay. Any questions about the vascular tree? Okay, so last thing we're going to talk about with the cardiovascular system lectures is blood pressure. Right, so blood pressure is the force exerted by blood against a vessel wall, and it differs throughout the vascular tree. Right, so arteries are under the highest pressure because they're right next to the heart. Veins are under the lowest pressure because they're farthest from the heart. Okay, blood pressure depends on the volume of blood in the vessel and the compliance of the vessel walls. Are arteries compliant? No, they're elastic. In response to being stretched, they recoil. What are our most compliant vessels? The veins. Okay. We're going to talk about blood pressure in two terms, systolic versus diastolic. Okay, systolic pressure is the peak pressure, right, exerted by blood when it's ejected from the left ventricle during cardiac systole. Okay, so when your ventricles are undergoing systole, when they are contracting, they eject blood, and that's the highest your blood pressure will be. And on average, during rest, for humans, it's 120 millimeters of mercury. Okay, so that's systolic pressure. Diastolic pressure is the minimum pressure in the arteries when blood is draining off. Okay, so this is when the ventricles are in diastole, when your ventricles are completely relaxing. Okay, this diastolic pressure on average for humans is 80 millimeters of mercury. Okay, and that's what's doing the forward moving pressure through the vascular tree. It doesn't go down to 0 like in the ventricles. It only goes to 80 because of that elastic recoil. Okay, so here we have arterial pressure going between 80 and 120. The difference between diastolic and systolic is known as your pulse pressure. That's the pressure you feel at your pulse points. Okay. So here we have blood pressure is increasing as blood is being ejected from the ventricles. Okay. Those semilunar valves slam shut. Pressure starts to fall. Oh, sorry. Here's where the semilunar valve slams shut. And I know that because that's where the dicrotic notch is. Okay, so ventricles are undergoing systole, ejecting blood. Ventricles start to relax. Okay, here's where the semilunar valve slams shut, or the aortic valve slams shut. There's that elastic recoil causing the dicrotic notch, and then blood flows down through the vascular tree. Pressure decreases until we get diastolic pressure. And then it happens all again. Blood gets ejected. This dotted line here is showing mean arterial pressure. Notice it lies closer to diastolic pressure than systolic pressure. It's not halfway in between. Okay? Mean arterial pressure is the blood pressure your body is regulating. Right? It's the blood pressure that it's detecting and regulating by altering systolic diastolic. Okay, blood pressure can be measured indirectly, right? The old-fashioned way with a stethoscope and a sphygmomanometer. Okay, there's electronic ones now, but in lab we make you do it the old-fashioned way. And when you're doing this blood pressure measuring, you're listening for the Kirchhoff sounds, okay? And those are the sounds heard, right, when you cut off blood flow, right, by increasing pressure above systolic. And then you can hear the little squirts of blood getting through. Okay, and then the last time you hear the little squirts of blood getting through, you know you've gone to diastolic because you got smooth moving blood. Okay, so this looks a lot better in this figure. So here we've got pressure readings. And then the green reading is the cuff pressure that you pump it up to, and then you slowly release the pressure. Right? And then here we have what those sounds are. So at first you don't hear anything because you've completely cut off blood flow. So you hear nothing in the stethoscope. coming through the brachial artery. Okay, the first time you hear a sound is because a little bit of blood squeezed through, right? And you can hear that turbulent flow. So that's systolic pressure. Okay, and then you're going to hear intermittent sounds as blood is able to get through. And then when you don't hear any sounds again, that's because cuff pressure has gone below diastolic pressure and you have smooth, continuous flow again. Okay, so pulse pressure can then be calculated, right? It is the difference between systolic pressure and diastolic pressure. Okay, so it would just be 120 minus 80. So the average pulse pressure is 40. And that's what you feel at your various pulse points. Okay, so if you want to determine your heart rate, okay, mean arterial pressure is what your body keeps track of. Okay, and we can estimate mean arterial pressure by taking our diastolic pressure plus 1/3 of our pulse pressure. Okay, so average diastolic is 80, pulse pressure is 40, 1/3 of 40 is around 13. So the average mean arterial pressure is 93 millimeters of mercury. Okay, so that is the pressure that's detected and regulated by your body. Okay, so here we've got pressure readings in the left ventricle. Notice that it's the highest pressure point and the lowest. It goes down to 0. Okay, and then here we have systolic and diastolic pressure in our large arteries. And we have this nice mean arterial pressure that lies closer to diastolic pressure. Okay, we still get pressure vacillations in the arterioles, right? But once we hit the capillaries, you don't get those vacillations in pressure anymore. Okay, you just have nice smooth flow. And again, fluids only move down pressure gradients. Okay, so you have to have lowest pressure in the venules and the veins. Right, so what we're going to talk about today to finish blood pressure is talk about blood pressure regulation. We'll talk briefly about short-term adjustments and longer-term adjustments. But we're going to focus on the short-term adjustments because we've already talked about the autonomic nervous system. Okay, short-term adjustments occur within seconds, so they're immediate. They happen really rapidly. Okay, and the body system that causes rapid effects is the nervous system. Okay, so the autonomic nervous system has influence on heart, veins, and arterioles. Okay, and the sympathetic nervous system in particular has big effects on cardiac output. Okay, and cardiac output is, what's the equation to calculate cardiac output? Heart rate times stroke volume. Okay, so sympathetic nervous system does what to heart rates? Increases heart rate. What does it do to stroke volume? It increases stroke volume. Now, stroke volume is equal to end diastolic volume. So that's the maximum amount of blood in the ventricles at the end of diastole, so the end of ventricular filling, okay, minus end systolic volume. So that's the minimum amount of blood. Remember, your heart can't squeeze out all the blood because it can't roll up like your toothpaste tube. Okay, so a little bit of blood always remains behind. What does the sympathetic nervous system do to end diastolic volume? Does it have an effect on end diastolic volume? Does it have a direct effect or an indirect effect? So what would increase end diastolic volume? Let's start there.
Decrease stroke volume.
So a greater end diastolic volume would increase stroke volume, right? If you fill the heart more full, it can pump more blood out. Where is that blood coming from that's filling the heart? Venous return, right? So if you increase venous return, you increase end diastolic volume. And what's going to increase venous return? Vasoconstriction. What causes vasoconstriction? The sympathetic nervous system. Excellent. Okay, so the sympathetic nervous system increases venous return by causing vasoconstriction of the veins. Okay, so the sympathetic nervous system increases end diastolic volume, right? Not by direct effects on the heart, but on effects of venous return. And then what does it do to end systolic volume? It decreases end systolic volume. And how does it do that? It causes the heart to squeeze harder because the sympathetic nervous system directly innervates the ventricular myocardium and causes it to squeeze harder. Okay, so the sympathetic nervous system has really strong effects on cardiac output by ramping up heart rate and increasing stroke volume. Okay, the longer term adjustments we'll talk about in more detail when we talk about the urinary system. Okay, these adjustments require minutes to days, and it's all about adjusting total blood volume. And your kidneys play a major role in regulating the amount and composition of your plasma. Right, so we'll talk about blood pressure regulation again when we talk about the urinary system. Okay, so in order to regulate blood pressure, your body needs to be able to detect your blood pressure. And it detects your blood pressure using mechanoreceptors that are in the carotid bodies. Okay, so you have these arterial baroreceptors in the carotid bodies. So they're detecting blood pressure of the blood going to your brain. Okay, and we maintain a really high blood pressure in the systemic circuit in order to fully vascularize our brain, because our brain's above our heart, right? So we want to know what the blood pressure is, in particular, going to our brain. Because what happens if the brain doesn't get enough blood? You die. It's true, you die. But what happens first? Before you die, you pass out. And what happens when you pass out? You lay down, right? You don't die when you pass out. It's something you die. That's not always the answer. Okay, you lay down and then your head is in line with your heart. Okay, so that it's easier to vascularize your brain. Okay, so those baroreceptors are constantly telling your brain what your arterial blood pressure is. So here we have normal. Notice our normal, for whatever reason in this figure, is set at 110/70. What do we normally say normal blood pressure is for humans? 120/80. For whatever reason, they decided in this figure to set it at 110/70. Okay, maybe this is for females. Okay, females tend to have slightly lower blood pressure because our heads aren't as far ahead above our hearts because we're shorter. Okay, so those baroreceptors are sending this baseline frequency of action potentials. When blood pressure increases, they send higher frequency action potentials. When blood pressure decreases, they send lower frequency of blood pressure, or frequency of action potentials. Okay? So those mechanoreceptors constantly detecting blood pressure, sending information to your brain, which is your integrating center. Okay? And then your brain is going to send out signals via the sympathetic nervous system. Okay, and that integration occurs in what's called the cardiovascular control center, which is in your medulla oblongata. So here's a cross-section of the medulla oblongata. And what part of the brain is your medulla oblongata? Who remembers? Brainstem. Okay, so is this conscious? No, this is completely subconscious. This is why when you want to know your heart rate, you have to calculate it, right? You don't just subconsciously know your heart rate. It doesn't send that information up to your consciousness. Okay? You have to use another sensory system to calculate your heart rate. Okay, so that cardiovascular control center in your brainstem is getting input from those arterial baroreceptors, okay, from these low pressure baroreceptors in your brain, which fire like crazy if they're not getting enough blood pressure, from chemoreceptors, telling you something about carbon dioxide levels and acid levels. Because why would you have an increase in CO2 or hydrogen ions in the bloodstream? What would cause that? You have an increase in hydrogen ions and CO2. What are you doing? Are you just sitting around?
Exercising?
You're exercising, right? You're doing a lot of metabolism. And what should happen to cardiac output if you're doing a lot of metabolism? It should increase. Okay. Right, proprioceptors. Okay. Your heart wants to know where your head is. If it's in line with your heart, blood pressure doesn't have to be as high. If you're standing up, it has to be higher. Okay. And then higher brain center baroreceptors. Again, your brain desperately wants to retain high enough blood pressure so that it gets fully vascularized all the time. Okay, the cardiovascular control center then sends out output to the parasympathetic nervous system via the vagus nerve. Okay, and long preganglionic fiber, short postganglionic fiber. And then it also sends out output, via the sympathetic nervous system, via the sympathetic chain, and it's going to send out input from the sympathetic nervous system to the SA node, just like parasympathetic nervous system, but also to the ventricular myocardium. Okay, so input to the SA node is going to affect heart rate. Input to the ventricular myocardium is going to affect stroke volume, right? Because the stronger the ventricles squeeze, the lower end systolic volume goes, and the higher stroke volume goes. Okay, so sympathetic nervous system innervates both the ventricular myocardium and the SA node. Okay, so it affects both heart rate and stroke volume. Okay, the sympathetic nervous system then also innervates our arterioles and our veins, and in general, sympathetic nervous system causes vasoconstriction or vasodilation. Vasoconstriction. Okay, and if you cause vasoconstriction of your arterioles, what happens to blood pressure? Increases. If you cause vasoconstriction of your veins, what happens? Increases stroke volume because you increase venous return, right? Which increases cardiac output. And an increase in cardiac output also increases blood pressure. Okay, so we have all these awesome mechanisms to increase blood pressure almost instantaneously. And what's our mechanism to decrease blood pressure short term? Slow the heart rate via the parasympathetic nervous system. Okay. Right? This is why hypertension is such a problem. Okay? We have loads of great ways to increase blood pressure and not as many ways to decrease it. Okay? Because your brain's way more worried about constant supply, right? So your brain is really worried about the here and now and not so worried about blowing out capillary beds, right? And long-term consequences of hypertension. Okay, so here we've got this nice table looking at extrinsic control of mean arterial pressure. So on the heart, extrinsic, remember, is always nervous system or endocrine system. Okay, so extrinsic mechanisms means outside the organ, and it's the nervous system or the endocrine system. So we already talked about the effects of sympathetic nervous system on both the SA node, increases heart rate, that's going to increase mean arterial pressure. Effect on ventricular myocardium is to increase the contraction strength, which increases stroke volume. That also increases mean arterial pressure. Now remember, in the sympathetic nervous system, it directly innervates the adrenal medulla and causes the release of epinephrine. So epinephrine's a hormone. Okay, so the endocrine system also increases heart rate and increases contractility. Okay, so you have the direct effect of sympathetic nervous system innervating your heart, as well as it releasing epinephrine from your adrenal glands. Okay, parasympathetic nervous system should decrease heart rate. I don't know why it's wrong, but it's wrong, isn't it? Okay, so if you printed out the slide, make sure you make this a down arrow. Okay, parasympathetic nervous system decreases heart rate. And that is what decreases mean arterial pressure. Okay. This is a really good review of what causes arteriolar vasoconstriction versus vasodilation in this segment of the table. Okay, so again, we have the effect of the sympathetic nervous system. In general, it causes vasoconstriction that increases total peripheral resistance. Remember, our arterioles are our major resistance vessels. Okay, you increase total peripheral resistance, you increase mean arterial pressure. Okay, epinephrine has variable effects depending on what kind of receptor that arteriole is expressing for epinephrine. Remember, there's all those different types of receptors. So the arterioles supplying our skeletal muscle express a different type of receptor, so that epinephrine causes vasodilation. Okay? Right? Other arterioles, especially just supplying like the GI tract, epinephrine is going to cause vasoconstriction. Okay. Two other hormones that we talked about, vasopressin, which is also known as antidiuretic hormone, and angiotensin II. Both of these we'll talk about in way more detail when we talk about the urinary system. They both cause system-wide vasoconstriction and therefore increase mean arterial pressure. Okay. And then the veins are also directly innervated only by the sympathetic nervous system. It causes vasoconstriction. That's going to increase mean arterial pressure because it's going to increase venous return, which will increase stroke volume, which will increase cardiac output. Okay, and then epinephrine also causes venous vasoconstriction. Right. I like to show this figure, right, because it shows that if you exercise, right, your sympathetic nervous system, even though it ramps up cardiac output, right, you don't see nearly as large of a change of mean arterial pressure as you would expect due to that massive increase in cardiac output. Okay, if you have your sympathetic nervous system firing because you're super nervous right before you take the exam about cardiovascular system, okay, without any exercise to go along with it, your mean arterial pressure is going to also skyrocket. Okay. So what happens is cardiac output increases because both heart rate increases and stroke volume increases in response to exercise. Okay? That stroke volume increase is going to increase due to an increase in end diastolic volume because you're going to have increased venous return. But you don't see as large of an increase of mean arterial pressure because all those blood vessels supplying your skeletal muscles vasodilate, right? And they allow the blood to drain off. So you actually see a decrease in total peripheral resistance. So even though the sympathetic nervous system is firing and causing system-wide vasoconstriction, right, the epinephrine will cause vasodilation of arterioles supplying the muscle, as well as those local chemical messengers. So remember, an increase in carbon dioxide and hydrogen ions and potassium ions and ADP, all those things that get released from cells that are doing a lot of metabolism, also cause vasodilation. Okay, so you really open up the spigots and allow all that excess blood from the increase in cardiac output to drain off into your muscles. Okay? So this is why exercise is really good for preventing hypertension. Okay? It gives an outlet to that increase in cardiac output. Right, the last thing we're talking about is blood pressure abnormalities, because you guys are all interested in going into a health field, and this is a major health problem. Okay, so hypertension, much more so than hypotension, right? But these are things that you're going to see clinically all the time. So hypertension, it's also known as high blood pressure, and it's clinically diagnosed when you have regular blood pressure readings above 140/90. Okay, and what's average? 120 over 80. Okay? Hypotension is low blood pressure, and this is regular readings of blood pressure below 100 over 60 that also show up with symptoms like dizziness. Right? So in lab, especially some of the small females might have had blood pressure readings around 100 over 60 and been totally fine. Right? But if you're 6 foot tall and you have a blood pressure reading of 100 over 60, you'd probably be dizzy. Okay, hypertension is the most common blood pressure abnormality. And the reason it's super common is because there's all these causes. Okay, so you can have defects in the salt management by your kidneys. So there's some people who are really salt sensitive. Okay, and so diets high in salt, if their kidneys aren't handling it correctly, lead to hypertension. Okay, so your kidneys are really good at reabsorbing sodium because it's our major extracellular cation. And we evolved in a low sodium environment. We did not evolve with salt shakers and processed food, right? So we used to have to be super efficient at retaining all our sodium. And that's why we have such a taste for sodium, okay? Salty food tastes delicious. Okay, because we used to have low amounts of sodium. Okay, imbalances in your ions can also cause hypertension. So diets low in potassium and calcium. Okay, so in order to reabsorb those ions, you're going to also reabsorb a bunch of water, increase blood volume, leads to hypertension. Okay, there are genetic defects. Some people have defective sodium-potassium pumps, and therefore retain an excess amount of sodium. Okay, some people have a variation in the gene that codes for the angiotensinogen, which ultimately converted into angiotensin II. And what does angiotensin II cause? We looked at it in the table. Vasoconstriction. So some people produce angiotensinogen that's easily converted to angiotensin II. Okay? They're much more sensitive to producing angiotensin II and therefore do more vasoconstriction. Okay, some people also produce more endogenous digitalis-like substances. Who knows what digitalis does? It's a drug. It increases heart rate. Right? So it's a drug that's used to regulate arrhythmias, okay, especially for people who have low heart rates. Okay, so people who produce a lot of endogenous digitalis-like substances have elevated heart rates. Okay, some people have abnormalities in nitric oxide production, endothelin, or other locally acting vasoactive chemicals. Either they don't produce enough, because remember nitric oxide causes vasodilation, or they produce too much and cause too much vasoconstriction. Right? And then some people produce an excess of antidiuretic hormones, so they reabsorb too much water, blood volume's too high. Lots of reasons for hypertension. Okay? Hypertension is a problem if it's chronic because it can lead to congestive heart failure because your heart has to work harder to push against that increased total peripheral resistance. Okay, so it works really hard and eventually it just gives out. Okay, it can lead to a stroke. What's a stroke? What organ is a stroke affecting? The brain. Okay, it either can blow out a capillary bed because of the hypertension, or you can get a blood clot that lodges. Both could cause a stroke. Okay, you can have a heart attack either because a blood clot lodges in the coronary circulation, or you blow out a capillary bed in the coronary circulation. Okay, if you blow out a capillary bed anywhere but your brain or your heart, it's just called spontaneous hemorrhage. Okay, it can cause renal failure. So those glomeruli in your kidneys, which are the fenestrated capillary beds where filtration takes place, If they're under higher pressure, you're going to get way more filtration, and then your kidneys are going to have to do way more reabsorption, and they just wear out. Okay? And then it also can cause retinal damage. So that increase in blood pressure can actually basically blow out the capillary beds in your retina. Right? And one sign of hypertension is when you see the floaters. Right? And if you don't know what I'm talking about, that's good. You see them all the time. Yep. Yeah.
So, would hypertension cause aneurysms, or is that different?
Hypertension can potentially cause aneurysms. That's true. Yep. So aneurysms are where you have a large blood vessel. So spontaneous hemorrhage is just small blood vessels sort of being blown apart. Aneurysm is a big blood vessel being blown apart, and that is bad. Yeah.
I'm seeing an example of that. One of the cadavers in the anatomy lab currently has a huge aneurysm.
Oh. In what blood vessel? Oh yeah, that's lethal. Yep. Yep. You don't want your aorta to bleed out. Okay. The opposite of hypertension is hypotension. So this is low blood pressure. It's either due to too little blood to fill the vessels. So severe hydration or severe dehydration could cause hypotension, or loss of blood, right? Or your heart is too weak to drive the blood. So heart failure, someone who's had chronic hypertension and then goes into heart failure may then end up with hypotension. Right? You have probably all experienced orthostatic or postural hypotension. So especially when you're sick. Okay, so if you had the flu this year and you spent a couple days in bed, right? And then you sat up too quickly, oftentimes you feel dizzy and you might black out. Okay. And this also happens when you vomit excessively and become super dehydrated. They're really worried about you passing out, right? And then circulatory shock is when your blood pressure falls really low, which either could be due to an excessive bleeding event or witnessing a traumatic experience, etc., so that you don't get adequate blood flow to your tissues. Okay. Right. Any questions about blood pressure regulation? Yeah, question.
So I'm still wondering how vasoconstriction in veins increases venous return, because don't you need those to be a low pressure environment in order to accept the blood?
That is true. So, but luckily, when you increase venous return and you fill the heart to a greater extent, it's the cardiac muscle, just like smooth muscle, in response to being stretched, it squeezes harder. Right? And when it squeezes harder, it increases ventricular ejection, and that increases blood pressure. So you're like, with an increase in venous return and venous vasoconstriction, yes, pressure in the veins goes up, but so does pressure in the arteries. Right? So they happen simultaneously. Okay? Right. Any other blood pressure questions? Right. Oh, another one. Yeah.
Yes, I have one more.
Yeah.
So vasodilation, is that purely driven by I can't remember what it's called, but blood cells releasing?
Local chemical messengers. So vasodilation, except for the arterioles supplying the genitals, which are innervated by the parasympathetic nervous system, that's the only exception, is due to local chemical messengers, okay, which then cause vasodilation. And those local chemical messengers trump the sympathetic nervous system. Yep. Okay. Any other questions about blood pressure? Right.